Population genetics is a fascinating field and also one of the most interesting things to come out of the modern synthesis. Let's talk a little bit about what that means. The name population genetics is pretty straightforward. We're studying genetics, we're looking at heredity, how traits are passed from offspring to from parents to their offspring, but now we're looking at the level of a population. So formally, population genetics is the study of the distribution and change in allele frequencies over time. So we're looking at an entire population and how these different traits will change and the frequencies of them. So let's go back to our definition of evolution. Remember, at its simplest, this really just means change over time. But now we have a specific type of data. So our definition now is change in allele frequency between generations. So we're looking at a specific trait and looking at the different variants or alleles. If you're thinking about eye color, you could look at how the number of people with brown eyes, blue eyes, and green eyes changes from one generation to the next. Of course, this is simply the population genetic definition of evolution. If you have different types of data, you'll have a different type of definition. Remember back to when we were talking about microevolution, when Peter and Rosemary Grant were looking at medium ground finches in the Galapagos. They were looking at beak depth, so they had a morphological measure, not allele frequency. So they couldn't use it in quite the same way we are. This is the simplest definition and most common definition of evolution that you'll find, but that's just because it's so concrete and easy to understand. Just remember that sometimes you might see different perspectives because you have different types of data and a different view to look at evolution. But let's look at the different measurements we're talking about here. Of course, the key word is frequency. There's a couple different frequencies that you're going to be concerned about in population genetics. First, phenotype, genotype, and of course, the all-important allele frequencies. So let's think of an example. The phenotype would be the people with brown eyes or blue eyes, whichever one you care about. But remember, phenotype is the physically observable trait. Then genotype, that would be the people with the genotype RR. And lastly, allele frequencies. That's the number of alleles, of our alleles in the population. This one's a little bit different and interesting because there are actually twice as many alleles as there are as people because every person has 2 alleles. So you want to make sure you're not off by a factor of 2. But let's go through an example for how we calculate these different frequencies. So here is a hypothetical table we have. So here we have people with brown eyes and people with blue eyes. We know brown eyes are dominant to blue eyes, so people with brown eyes would either have genotype RR or big R and little r, and people with blue eyes would have 2 recessive alleles. So we already know the counts for the different genotypes here. Here, our population size is simply all the people we have, which for this example is 24. It's a pretty small population. First, we want to calculate our phenotype frequency. So we take then everybody with brown eyes, we add them together, we divide that by 24. We have about 80% here. Our genotype frequency, now we're looking at the genotype for big R. So we just have 8 individuals, we divide that by 24, and we have a third of our population here has that genotype. Now our allele frequency, and now we want to start paying a little bit more attention. Here, you notice it's a little different in our denominator. So now we're looking at the frequency of the big R allele. Well, 8 people with the homozygous dominant genotype have that, so we have 8. But we multiply that by 2 because those individuals have 2. Then we add 11 because 11 individuals have 1 big R because they're heterozygotes. And then we divide that by 24 times 2 because that's the total number of alleles in our population. So always be careful that you make sure to times your population by 2 when you're calculating an allele frequency. Of course, because frequencies always have to add up to 1, if you want to figure out the frequency of the other allele, you just do 1 minus your previous number. So don't forget that handy cheat. So can you explain? What is population genetics and what do you study in this field?